Welcome to your vodcasted lecture for Monday, March the 10th. This is the second of 4 vodcasts for the period of time that I'm gone with the ski team at their national competition. And if things have gone really well, then I should be safely settled in New York, and we should be all prepping today for the first race, which will actually happen tomorrow. And something happens for you all tomorrow as well. Homework 6 is going to be due, so you can be looking forward to that, but even more so, you can be looking forward to meeting with me from New York online. I will be available tonight for my usual 7:30 to 8:30 office hours on the conferences tool on Canvas. I will also be available 7 to 8 p.m. on Tuesday night for another office hour. So you guessed it, before tomorrow's due date of homework, you can meet me not once, but twice. So find me online at both of those times if you would like to log in and be a part of the office hours during those times. Do bear in mind that I will not be in my office for the 2 to 3 p.m. office hour that I normally hold on Monday. So the couple of you that usually meet me there, remember that I am definitely not going to be there. You've got a couple of really rad things coming up in lab. Obviously your skills tests, and those need to be completed. You will only have one attempt on each skill this week to get finished off with those rubrics. Those will need to be submitted to your TAs no later than whatever is your first lab on the week after spring break. That is the Tuesday of spring break. You're going to need to hand those over to your TAs. So we've been working on that for a while, but we're going to be starting something really, really rocking and brand new. And that is that you're going to be getting some bacterial unknowns, and we're going to have you identify those unknowns. And towards that journey, the first thing that you're going to get is a set of clues. And these clues are going to help you out in making a hypothesis as to the identity of your unknown. So you're going to want to go through the assignment description, and your TAs will also go through that with you, but you're going to want to also think about the process that you're going to need to use to identify these unknowns. And so we're going to have you write a flow chart or a description of the biochemical tests that you're going to do to try to identify each of these unknown bacteria. So that conversation will be had in your lab as well, but I wanted to give you a little bit of foresight into that assignment and the up and up on the very sexy-minded process of unknown identification. So look forward to it. This is your foreplay for the actual event, but this is something that you're going to need to start thinking about, and you're going to be journeying towards that unknown identification. That being said, I don't know about you guys, but I feel like I have literally studied translation 5,482,052 plus or minus a couple of times. So if you feel like you have studied translation that many times as well, guess what? Today we are totally going to spice it up, because I have brought with me a couple of rad things. That is to say, I just felt like wearing my ribosome t-shirt. So I'm wearing my pretty rad ribosome t-shirt. You can see my tRNAs and my ribosomal subunits. So that's pretty cool for one thing. But I also have some other toys that I've brought with me. I've got a couple of amino acids. These are going to come into the picture. I've got, well, heck, I even brought a giant amoeba with a winter sweater, a nice winter scarf on. So I think we should name him Rex, actually. I also have a squirt gun with me. It's a little tiny squirt gun, and if you stop paying attention to this lecture, I am totally going to squirt you. Either that, or when my cat comes and interrupts, I'm going to squirt her. So either way, this is a win-win situation with the squirt gun. Okay, good. So that means we are poised and ready to start the process of translation. Remember our big picture, central dogma of gene transfer. We had just talked about the process of transcription, whereby RNA polymerase converts DNA into RNA, and that RNA might be one of 3 types. It might be mRNA, tRNA, or rRNA. mRNA is the only kind of RNA that then gets translated. That is, it is read by a ribosome and converted into polypeptide chain that folds into protein. However, what's really cool is that both tRNA and rRNA are the workers of the process of translation. That is, rRNA is a big part of the ribosome that builds the polypeptide chain, and tRNA actually serves as an adapter, adapting the mRNA code to the polypeptide chain. So whereas tRNA and rRNA are the workers of translation, mRNA is the subject of translation. That is, it is what is converted into the protein core. the protein code. Without further ado then, let's go ahead and talk about the nuances, the details of the process of translation, beginning with the subject of translation, the mRNA. This is zoom in, up close and personal on the mRNA. And this mRNA has three distinctive regions that I have made three distinctive colors. Notice that there is an upstream untranslated region, and there is also a downstream region that is not translated. So these regions actually turn out to be pretty important on the mRNA. Maybe you can think about some of the reasons that these areas might be pretty important to the mRNA. For example, in the non-translated leader sequence, you might guess that this is where the start line for translation is. Remember how we had a promoter that was the start line for transcription? Well, translation also needs to have a start line, and it's inside of that non-translated leader sequence that all of the ribosomal equipment and whatnot gets ready for translation and finds its start site. Now, the non-translated trailer region down here is also super important. Picture, for example, a nuclease enzyme. Remember that nuclease enzymes super love to chew up nucleic acids, and mRNA is particularly subject to their digestion. So if we literally picture a Pac-Man enzyme coming up and having at the 3' end, so we've literally got Pac-Man here, chowing down. Did any of you guys, you're way too young to have played Pac-Man when you were little, but if anybody out there did play Pac-Man when you were little, we've totes got Pac-Man chewing down the 3' end of the non-translated trailer region. Now, you can automatically see the incredible value of a non-translated trailer region when you realize that this Pac-Man enzyme can chew and 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 chew, and it doesn't ever reach the important part of the mRNA, that is the coding region of the mRNA. So the non-translated trailer region protects the coding region of the mRNA, and it allows that coding region to stay untouched by Pac-Man nucleases in important times. So that coding region can be fully and appropriately expressed to form protein, and it is the region that is converted into the polypeptide sequence. Now, I'm hoping that everybody is actually visualizing already the form of a protein as a polypeptide chain comprised of the 20 standard amino acids. But just in case you didn't get that amino acid table in the right place and you're not staring every single day at amino acids, then let's remind ourselves of the basic structure of an amino acid, recalling that every amino acid has a central carbon, it has a hydrogen, it has a carboxyl, and it has an amino. So let's get them right here. Amino on this side, NH3 plus carboxyl on this side, C double bond O minus. And then of course it has an R group. That R group varies, there are 20 of them. And we recall all of their different natures. So when we think about this amino acid structure, recall that as an mRNA gets read, that mRNA encodes for amino acids. And as more and more of the mRNA is read, more and more amino acids get incorporated into the ever elongating chain of the polypeptide. So notice that the next thing that's going to happen is that, and Kelvin will remember this, I think you were the one that I handed the little balls to, when the next amino acid is added on here, then we'll get the formation of a peptide bond. This will bind amino acid 1 to amino acid 2 with its quintessential amino acid structure, its R group, its amino bound to the carboxyl of the first, forming that peptide bond. So what we notice is that as amino acids are added to an ever elongating chain of amino acids, always this chain, always is built amino to carboxyl. So always new amino acids are added onto the carboxyl end, in the same way that always new nucleotides are added onto the 3' hydroxyl when building DNA or RNA. So proteins always built amino 2-carboxyl. Now, just in case you're like zoning out and you're stopping paying attention, I've totes got the squirt gun, so don't make me use it. But I've also got the amino acids, and we're going to look again at the model's 3-dimensional space, always very aesthetically pleasing. So let's go ahead and see that bond being formed between the amino groups. So let's make sure that this is looking right from your angle. So amino 2-carboxyl. Okay, I think it's a little good. So we are going to cleave out the little balls, and we're going to form the peptide bond. Again, making sure that everybody remembers how that peptide bond is formed. And we'll try to get it right here. Aha, epic. Peptide bond formed, amino 2-carboxyl, and you guessed it, the little balls that comprise water. Excellent. So, nice review. Hopefully you're set and you're poised to understand the rest of the process of translation with a really visual element of having seen the amino acids building. So, during translation, amino acids are added, as I said, to the carboxyl terminal, making an ever-elongating chain amino 2-carboxyl. But what is even more important to remember is that the mRNA is what's dictating what amino acid goes next. That is, whether or not leucine or isoleucine or threonine gets added next onto the elongating chain is very much dictated by what the mRNA sequence says. So the mRNA is the code dictating the amino acids. But we have a problem. We have a big problem, in fact, because the number of nucleotides doesn't match very well with the number of amino acids. That is to say that in RNA there are only 4 nucleotides, right? We can count them. There's U, there's G, there's C, and there's A. But that's not enough to dictate amino acids, right? 4 nucleotides versus 20 amino acids, something doesn't add up real keenly. And so you might say, well, doesn't that mean that maybe you could read 2 nucleotides and then those 2 nucleotides could specify the amino acids? But when you think there are 4 nucleotides, if you square that, that's not going to give you enough either, right? 16. So that's not quite enough to give you the 20 amino acids. So that means you actually have to use 3 nucleotide sequences, and these are given the name a codon. So 3 nucleotide sequences are read one at a time by the ribosome to make that elongating polypeptide chain. My shirt shows it, so let's check it out. Notice you can see the ever-elongating polypeptide chain going up my shoulder, and you can see the ribosome is what's spitting that out. And all the way down around my waist, maybe you can check this out, there is the 5' to 3' RNA. So, pretty rad. This is how these are read. 3 at a time, so we can go 4 nucleotides read in 3 nucleotide per codon sequences is 4 raised to the 3rd. And if you're doing the math on that, I'm positive that Ben has already done the math, and you know that you get 64 codons. 64 codons. Something doesn't seem quite right here either, because remember that there were only 20 amino acids. 64 codons to dictate 24 amino acids. What does that mean? I know, you're probably yelling at the screen, but what this means is that a codon, there must be more than one codon to specify a single amino acid. And this is actually, it's given a very sexy term. It's called degeneracy. So degeneracy literally means that there's more than one codon that can dictate a particular amino acid. And the genetic code must be pretty darn degenerate, because in fact, there's going to be oftentimes 3 or 4 or 2 different codon sequences that dictate the same amino acid. Also, there are some codons that don't dictate an amino acid at all. And in fact, what they dictate is a stop codon. So let's go ahead and look at the key that we can use to allow us to determine what a codon, what amino acid is incorporated based on the codon. This is called a codon usage table, and firstly, it shows us that there are 3 codons, UAA, UAG, and UGA, that specify absolutely no amino acid. That is, they are stop codons. So when a ribosome comes across a stop codon, it's going to be like, whoa, I don't know what to do at this point, and there's no tRNA that specifies this particular codon, so the ribosome has to stop at that point, and this leads to the ending of the process of translation. But that still leaves 61 of the 64 codons that specify unique amino acids, or I should say a particular amino acids. And so that means that we have more than one codon specifying the same amino acid. Nothing could show that better than this codon usage table. For example, if you look in the lower right-hand box, you can totally check out the fact that there are 4 different codons that are listed just in this lower right-hand box that all specify glycine. So remember glycine, that tiny little amino acid? It's pretty rad because it's totally flexible. It fits into really small spaces. I know some people like that. So GGU, GGC, GGA, GGG, all of them specify glycine. That is degeneracy at its best. That's not the only amino acid that has more than one codon that specifies it. And in fact, look at alanine over here with GCU, GCC, GCA, and GCG all specifying alanine. So let's talk about some of the others that are used to specify particular amino acids. One very, very important codon is AUG, and AUG signals go. That is, it is always what begins the process of translation. Translation always begins with an AUG. And so wherever that first AUG is found past the start line is where the process will begin to incorporate amino acids. So it's where translation will begin. In prokaryotic cells, the particular amino acid that's specified by AUG is something called N-formyl methionine. That is simply because there is literally a formyl group attached to the amino terminus of this methionine residue. So if we picture that on the N of the amino terminus, there is actually a C double bond O bond H. That is, this is a formyl group on the amino terminus of the first methionine. So in prokaryotic cells, this is not just methionine, it is F-met. So that doesn't fit real well, but N-formyl methionine. Let me write it out here. This is F-met. And in case you're having trouble with visualization, please do remember that this goes on to the central carbon and the hydrogen and the R group, right? And the amino acid that's here. So this is a formulated end, and that is always how the process of translation begins in all bacterial cells. Okay, let's go ahead and look at some of the other codons and what they specify. And I can totally tell that some of you are falling asleep. Don't forget, I have my squirt gun and I am not afraid to use it. Okay, so let's look at some of these other amino acids. And firstly, UAA, UAG, UGA, we already talked about how they specify stop. But look at the degeneracy that we also see in the second column. UCU, UCC, UCA, UCG, all of these specify serine. And of course, we can also say that there's degeneracy for our favorite amino acid, histidine. CAU, CAC, both of these specify histidine. So degeneracy is very prevalent. And maybe by now you've started to get the feel of the location in which the code is degenerate. That is to say that it seems like the total loosey-goosiness is happening in the most right-hand side of this codon. Well, remember that nucleic acid sequences are always written in the 5' to 3' direction. So the loosey-goosiness of a codon, said another way, is in the most 3' position of the codon. The 3' position is the position that can change without changing the amino acid that's specified, or at least only changing the specified amino acid a little. For example, look at GAU, GAC, GAA, and GAG. GAU and GAC specify aspartate, but change just slightly in that 3' position to glutamate. Glutamate, remember, is the other acidic amino acid. So both of these functionally are fairly equivalent. That is, they have the same chemistry. They're both acidic. They both have the carboxyl group on their R group. So we see that that third position is the position of most flexibility within a codon. But I think we better practice, make sure everybody is all set to go and understands how to use this table. So just really quickly, what amino acid is specified by the codon CGA? So I'll give you a second. The codon CGA. Well, the way you use this table is to first find the first position on the far left, C. Then you find the second position on the upper area, G. And then lastly, you find the third position on the right. So CGA, ARG, right? The pirate amino acid. Oh wait, I totally got to show you guys a cartoon. This is absolutely hilarious. Give me one sec. I've got it right here on my desktop. Pirate amino acid, it's called. It's just hanging out there for this very moment. Oh yes, there it is. ARG. So remember that the pirate amino acid, arginine, and here, like, I love this. Here you've got the ribosome looking like a pirate with its eye patch headed down the mRNA, reading the mRNA in the 5' to 3' direction. And because it's a pirate amino acid, it's incorporating a polyarginine. So the only amino acid in the polypeptide chain is arginine. So yeah, totally a great cartoon. And I want to take a minute now to talk about the fact that not only do codons, and specifically the start codon, AUG, not only does it encode for an N-formal methionine, but it also serves to start the reading frame. So what that literally means is that the ribosome begins reading in triplicates no sooner and no later than the AUG start codon. So the beginning of the process of reading in triplicates always begins with AUG, because AUG literally specifies the right reading frame. Let me show you what I mean by that. When we look at this mRNA traveling 5' to 3', notice that the first thing that you come to, or the first important thing that you come to as you read 5' to 3', is that AUG start codon. So the AUG start codon sets the reading frame. And what that means is that that is the first codon that will encode for an amino acid. Now the second codon is CGU, and UCG is the third. It's so essential that you begin reading at the first AUG. If you don't, your reading frame is completely botched. So if you began reading, for example, right here at CGA, and you said, oh yeah, look at me, I'm totes a good ribosome, and you're reading on down this mRNA, notice this. Yeah, turns out you're totes a bad ribosome. Because notice you have a complete reading frame shift, a complete error in the reading frame. Whereas if you start, as you should, with the AUG start codon, then the reading frame is correctly and accurately set. So make a note to yourself, maybe next to the tattoo that you're planning to get that says 1,000 microliters equals 1 milliliter, maybe you should also get AUG sets the reading frame, because that is so very important. In fact, all genetic code would be completely botched if the ribosome didn't start at the AUG start codon. So it's absolutely essential to all of the phenotypes that we express that AUG sets the reading frame. Very nice. Let me know if you get that tattoo. As we begin our process of translation, the first thing that we need to do is to introduce the players. Ribosomes are clearly one of the most key players in all of this process. Ribosomes are the factories of translation. They're the ones that are literally moving along the mRNA, reading the mRNA, and enabling that mRNA to be converted into protein product. And enabling that mRNA to be converted into protein product. So scanning along the mRNA, reading the codons in the 5' to 3' direction, and presenting those codons in order to then adapt them to become polypeptide chain. Now, it's very important to note that the RNAs inside of the ribosome are literally the enzymatic function of these ribosomes. That is to say that the RNAs are serving as ribozymes. They have catalytic activity. So where there may be 15,000 to 20,000 ribosomes in a growing E. coli cell at a given time, you can imagine a lot of ribozyme activity. And if that turns you on the way that it turns me on, you might just want to go find some lectures and some publications by a man named Thomas Cech. Thomas Cech was a professor at CU Boulder, and he was one of the first to discover that RNAs can also have enzymatic activity. So remember that we tend to associate proteins with being enzymes, and we tend to think of them as being the movers and the shakers and the workers of the cell. But it actually turns out that RNAs can also be catalytic, and within the ribosome, the 23S ribosomal RNA is literally what provides the enzymatic activity, the ribozyme activity that facilitates the joining of amino acids. That is, it is what is making the peptide bonds. So within our prokaryotic ribosome, within the 50S or large ribosomal subunit, there is this epic 23S rRNA. And this 23S rRNA is what is acting to build the polypeptide chain. It is synthesizing peptide bonds. It is what is responsible for the production of that polypeptide chain. And don't be mistaken. It's not just the RNAs within the 50S subunit that play a huge role in the process. Also in the 30S subunit, there is an absolutely incredible rRNA called the 16S rRNA. The 16S rRNA serves to align the Aug Start Codon in the right place. So it aligns the start line for translation and puts the Aug Start Codon in the perfect place to allow it to set the reading frame. So the 16S rRNA is absolutely essential to proper alignment of the mRNA. So within both the 30S and the 50S ribosomal subunit, you have both RNAs and polypeptide chains, and they are all very important to the process of presenting the mRNA codons to allow those mRNA codons to encode for the amino acids that are incorporated. So not only is a ribosome a player in all of this, and of course remember that the rRNA, very, very important, but remember too that the tRNAs are also workers in this process. So without further ado, let's introduce the tRNAs. Oh, and by the way, pay attention, I will still have my squirt gun in the next episode.